Good morning, dear students. In this class, we are going to discuss on the isolation and identification of natural products. So, what are natural products? So, natural products are application-oriented molecules that are derived from organic sources, particularly that are extracted from plants, and that is what called natural products. So, how can we extract? How can we isolate the biologically important molecules from the natural sources? That is what called isolation of natural products. So isolation means separating the desired compound from natural sources. So there are different steps in separating the desired compounds from natural sources. So the following are the general steps in the isolation of an application oriented compound from natural sources. The first step in the isolation of a natural product is collection and preparation. So first, the plant or microbial source, natural source for the intended compound should be selected. So dry the plant material in shadow and finely powder. So first of all, we should find out the natural source, any specific plants or microorganisms from which we can extract the application oriented molecule. So once you select that particular plant, you just collect the plant in large number and prepare the particular source. So how can we prepare that? First of all, we need to dry the plant material in shadow, and once it is completely dried, it should be finely powdered so that the extraction process will be more effective. So if needed, the fermentation is carried out for microbial products. So if the source is microbial, in this case, fermentation is needed. If it is some plant material, then complete dryness and finely powdering is needed. Right? So it depends on what kind of source it is. So this step is called collection of source and preparation of the source for the extraction. Right? That is the first step. The second step is extraction is a very crucial and important step. So it is the removal of intended compound using sustainable solvents is called extraction. So extraction means the removal of intended compounds using sustainable solvents, right? So extraction process is commonly carried out using any of the methods like maceration, succinate extraction, steam distillation, liquid extraction, etc. So extraction is a very common word. So there are different methods are used for the extraction process. So extraction is actually removing the desired molecule from the natural source, right? So this extraction depends on the source and depends on the molecule we are going to extract. So different methods are generally carried out. So the most important methods are succinate extraction, maceration, steam distillation, liquid extraction, etc. So polar solvents like water, ethanol, methanol, etc. Non-polar solvents like hexane, chloroform, etc. are commonly used for the extraction. So mostly the solvent extraction methods used in succinate extraction or liquid extraction, etc. So both polar and non-polar solvents are generally used for the extraction process. So polar solvents, example, water and ethanol, methanol, etc. And non-polar, hexane, chloroform, etc. So these are generally used as solvents for the extraction. So based on the molecule and the source, we can select any one or more solvents from this list. So the third part, once we extracted it, so what is the next process? Fractionation. So what is the fractionation? So fractionation is the separation of crude extract into fractions based on the polarity differences. So acid base extraction or solvent partitioning method are used for the fractionation. So fractionation is the crude extract is actually a collection of different molecules. So among the different molecules, one is our desired molecule. In this case, we need to fractionate the crude sample into different groups of compounds, and that process is called fractionation. So acid base extraction or solvent partitioning methods are generally used for the fractionation process. And the fourth one is purification process. This is another important step in the extraction of a natural product. So purification means we just partitioned or we just fractionate the crude sample into different groups based on the nature, functional group, or acidic nature or basic nature. So based on its property, physical property, we just extract the crude sample into different groups. So from that group, we need to isolate the desired compound completely, and also we need to purify the desired compound, and that process is called purification. So actually in this step, we get the pure compound which is needed. So the compound is finally purified using a recrystallization or chromatographic method. These two methods are generally used for the purification or complete isolation of the desired product. So one is recrystallization, another one is chromatographic method. So what are the chromatographic method? Usually column chromatography, thin layer chromatography are used as a chromatographic method. So chromatographic method is actually separation of compounds based on their polarity, based on their diffusion nature in the solvents of different polarity, right? So it is a well established method for the separation of a mixture of compounds into uh, pure compounds, right? So TLC is generally used to identify, and there is one more TLC is there called preparative TLC, where you can also isolate the desired compound when it is in small quantity. And if the quantity is more, then we need to go for HPLC, high performance liquid chromatography. So once we purified the compound, so we get the compound. So we just isolate a specific compound from a bunch of compounds present in the natural product, which are first extracted, and then it is partitioned, and then finally it is purified. Now we have the pure compound. So the next step is very important step, that is identification. So what molecule we get? So we need to structurally confirm the molecule. So that is what called identification of natural products. So isolated pure compound from the natural sources are structurally identified. So what are the different methods used for the structural identification? So it is very similar to the analytical method we are used in lab. Right? So first one, we need to go for the physical properties of the compound. So physical properties like color, odor, melting point, boiling point, and the solubility of the compound are initially analyzed. So this is the first step. Next, the chemical tests. So based on the compound, we need to conduct certain chemical test, and thereby we can identify the compound. So specific chemical analysis to find the functional groups is carried out to preliminary identification. Say for example, there are so many examples out there for a specific test for particular group, like alkalides, Legendre test, phenols, ferrichlorate test, terpenoids, Salkowski's test, flavonoids, Shinoda test. So these are some examples. Similarly, for every particular functional groups or particular compound, we have a specific test. So by conducting the specific test, we can identify that particular functional group or that particular group of compound present in the isolated molecule, right? So these are all just preliminary tests. The major source or major methods of identification is spectroscopic methods. So there are different spectroscopic methods are there. So using this method and interfering the spectroscopic results, we can exactly identify what is the three-dimensional structure of that molecule. Some of the most important modern techniques used in analysis an unknown compound or UV visible spectroscopy. So using this, we can find out the conjugation present in the molecule. So IR spectroscopy, we can find out the functional groups present in the molecule. NMR spectroscopy, structure, proton environment, these kind of things we can identify using NMR. So the different NMRs are also there, proton NMR, C13 NMR, P31 NMR, F19 NMR. So using all this NMR spectroscopy, we can almost certainly confirm any particular compound. And mass spectroscopy confirm the molecular weight, And finally, there's another important method is that called single crystal XRD. So once the compound is crystallized, isolated and crystallized is in the pure nature. So using that crystal, we can analyze single crystal analysis. So the single crystal XRD analysis exactly give the three-dimensional structure of the molecule. This is the ultimate method for identification of the particular molecule, right? So the extraction and identification of natural products. That is what we see. So the last method is the one more method is there. That's comparison with standards. So once you identified that compound, you can also confirm that compound by comparing it with another standard. Say for example, if you suspect that particular molecule is any specific phenol, say for example, some phenol like pyrogalal. So then what we can do, we can get the commercially available pyrogalal and compare the compound with pyrogalal using TLC or gas chromatography, right? So this comparison also no need to go for difficult spectroscopic methods. The simple comparison is enough here, right? So once the compound is already known, means we don't need to go for a different spectroscopic method to structurally confirm the molecule again. Instead, we can also compare that molecule with another available source like commercially available original product, right? So using that we can compare the natural product and thereby we can confirm the molecule. So this is another method, right? So that's all. Thank you.